So it is AI driven, AI driven tumor classification from histopathological images. So now we will look at what histopathological image is. You should know that. You have seen these type of slides in many textbooks. Yes, these type of pink stained, like muscle cells, even in school books you have, right? These type of stained images. So these are histopathological images. So let us first break down what do you mean by this word histo and then pathology. So histo relates to a word, histo means tissues. So study of tissues. So histo means tissue and pathology when you study diseases. Okay. So histopathology is study of diseases by observing tissues under microscope. So when we observe these cells or tissue level images, these are transparent and you want to look at it. So you have to stain them. Okay. So particularly in histopathology, mostly used stain is H and E stain. Please, you should note it down. Okay. Why? Because even if you're not noting it down now, if you go through the recorded video later on, just note it down. You must know these things. So this is known as hematoxylin, okay, and eosin. So we are using two stains. Anyone, can you tell me when we use more than one stain? So this is a wet lab question, okay? So wet lab biologists, you must tell me, if we use more than one stain, what is this process known as?
The differential staining?
Correct. This is differential staining. See, this is the basic, you learn in microbiology or biotechnology, whatever you take, you either in first or in second year, you in microbiology lab, you as a biologist, you should learn this, and you do learn this. That's in the syllabus, okay, in almost all the colleges. So this is a differential staining where we are using more than two stains. The first is hematoxylin. Now the color is blue or purple, okay, and eosin usually we perceive as pink, but it can be sometimes red also. So eosin, the word eosin, mostly it is related to red color, but since it's diluted we perceive it as pink color. Mostly it's pink in color, right? Now let me proceed. So what you will understand after this page or this class today is that what a histopathology image is, how it is produced, and now whenever it comes for cancer diagnosis, this image is gold standard. Not X-ray image, not MRI image, but histopathology image is the gold standard for cancer diagnosis. Okay, why we will look at it later on. Now we can also see biological hierarchy in the tissue slide. Now, what do you mean by biological hierarchy? It will follow further in the lecture today. So stay attentive. What do you mean by hierarchy? Now, hierarchy means levels. So we can visualize these slides in different levels. Okay, just remember that. And now it will also show some visual differences between three types. Now, the tissue can be normal tissue with no disease. Now, if suppose there is a disease, it can be either benign tumor or it might have metastasized and it can become cancerous. So, when the cell is cancerous, it's known as malignant. Now, can anyone tell me? I think one more person, he has not joined yet. Okay. So, can anyone tell me what is the difference between cancer and tumor? So, when I'm saying you tumor, this can be known as benign. And when I'm saying cancer, this is known as malignant. Can anyone say, or shall I? Anyone knows about it? Any slightest of idea you have what is difference between these words? Now both are diseased. Both are diseased, not a normal condition, both are diseased condition. So when cancer, everyone, we have gone through this word. We have stages of cancer, stage 1 to stage 4. So in the final stage, what the cancer cell does is it leaves its location, okay, known as mesenchymal to epithelial, M to E transition. So this is something biology, deeper biology, cancer biology. So it transits. So it will leave its, local area. These cancerous cell, they will rapidly divide, and in the last stage, it will leave it area and spread throughout the body. At that time, this is known as cancer, or it is known as malignant. Before that, so from stage 1, stage 2, stage 3, there are so many different changes, biochemical changes that are happening, and the cells, they are becoming, tumor cells are becoming more invasive, stage by stage or day by day. But they do not leave their location, they are localized. So the treatment becomes easy. Okay, easy treatment. But when it metastasizes, it becomes malignant, it becomes cancerous, then it becomes systemic. Please, if you do not know these words, please just grasp it now. Systemic, full body, it is going throughout the body, it's circulating throughout the body. So this is systemic. I hope now you understood what is normal, benign, and malignant. Yes, if you have any doubt, you can ask me, I'll repeat it, and only then we'll proceed further.
Explain again, ma'am.
Sorry?
Once again, ma'am.
Once again. Okay. Now, I have told you there are 3 cases. First is normal. Your cells are normal. Nothing is happening. So when, suppose if a patient is, due to any, there are several reasons for cancer. Okay. So cancer, you don't get suddenly. The first stage, maybe there are some mutations. Okay. DNA. So that DNA, there are some mutations in the DNA. Fine. Due to that, what happens, the cell, it divides fast. So there are some signs of cancerous or tumor cells. First of all, cancer means nothing else but it divides fast, and not only fast, it divides uncontrollably. Now, and then there are so many, change in shape, change. You will. I'll tell you what changes are there. Okay. So in this lecture itself, you will come. You'll go through there. Go to. Sorry. Go to. So sorry. Go to those changes. So there are normal cells, and then you have got. In initial stages, you are. It is known as benign. So at that time, when the cells are benign, I mean they have developed into tumor cells. Okay, their DNA is haywired. I mean, what do you mean by haywire? It goes crazy. There's no control, fast dividing. Okay, and it does not only affect itself, but it will start affecting their neighbor also. Its shape will change. Okay, so there are so many things which I cannot explain in one single go. Cancer biology is a vast field for you to understand. So this thing, nothing happens in normal cells. If suppose there is a damage also, it will undergo self death, apoptosis. Okay, but in when the disease has just started and it is turning into a benign tissue, at that time what happened, the normal cell, uh, it is benign. Okay, now, so this is the second stage, benign. Stage 2, stage 3, stage 1, at that time, if it is not leaving its own place, and if it is not traveling or not spreading throughout your body, it is known as benign stage. At that time, you can say it is a tumor. It is an uncontrolled growth of cell. Growth of cell. Sometimes it is not harmful. You can just cut that portion out. Okay. But what happens, like suppose this is the cell. there is a mutation. I hope everyone knows what is a mutation. So there is a change in the DNA sequence. Now, when it divides, it will pass on the mutation to its daughter cells. Then there will be twofold mutations, because once the cancer cell has been, marked that it is a cancer cell, now nothing will go right. So it will not stop with the first mutation. This cell will have more 3 to 4 mutations, this cell will have more mutations. Now these both will not be same. You don't think so that if suppose in this patch of skin here there are so many cancer cells, all cell will be different from each other. Do you understand? Because they are fastly dividing. Two daughter cells, they both already they have got mutated DNA. Now they are transitioning into tumor cells. But new what I can say, it is also undergoing new mutations and new changes, their own changes. So what it does, it becomes a cancer mass, a tumor mass is always polymorphic. No two cells may look identical, but when you see a normal tissue, most of the cells will be identical to each other. They look quite similar, but not in tumor cells. Now, when these tumor cells, okay, when they have got the ability to leave their place at a tissue level, the cells are located, these dots what I'm drawing, suppose they are cells. Okay. Now these are tumor cells only. What they do, they move from their membrane, from their place, from their basement membrane. If they move from that place and spread throughout the body, at that time you say that it is a cancerous cell, and it will look much more disgusting as compared to a tumor cell. Did you get a little bit of idea what I'm trying to say? It will look very bad. The cells will not look normal. And it is a very complex process, not a one step, two step process. It is a very, we can say there are 4 stages of cancer, but it is very complicated. Understood? So when do you say cancer or malignant? When it is spreading throughout, it is leaving its own place and spreading throughout the body. That is cancer, and it is very difficult. At that time, whatever medicine or whatever treatment you are taking, it must be systemic. It should target whole body. Whole body when it is targeting, that is why you have got hair loss. You have got a person undergoing cancer treatment, their immune system is down. So everything is being attacked by the drug, the systemic drug. Systemic means overall. It will not only attack the cancer cell, but it may also attack the healthy cells. Okay, so this was the difference between normal, benign and malignant tissue. Now we are working with AI. So what AI does, AI is nothing but we are using computers. So computers will scan them. How? Any image is divided into small dots known as pixels. So it should read those pixels, and with the help of those pixels it will predict. Okay. Now histopathology is very complex. It is not a normal image, it is a complex image. Okay, we have got 2 dyes, 2 stains. Then we have got so many patterns. Normal cell will be very, okay, let me just show you now. Okay, this one, you see? So the normal cells pattern is so, they are arranged so neatly. So these are glands. But the same thing, you see, the glands have disappeared. There's no structure, nothing. So these patterns also, apart from pixel, color density, edges, pattern, all these things, your model, your AI model should be able to get it and distinguish between normal to benign or benign to malignant. Understand? So how do you do this? With the AI will just look at the pixels. I'll tell you how the pixels are converted into mathematical numbers. I'll tell you later on. So how to observe a slide systematically? Before you teach your computer, again and again I'm telling you, you must know. So there's a 3-level scan protocol. We will just look at that later. So 5 most common beginners mistake and how to avoid them, that also we will do. Now you know, for all these steps, a doctor, pathologist, okay, he goes years of training, only then his eyes are trained, and then it is not so simple. I just showed you a ultra magnified and AI generated image over here. Okay, but no, the real histopathological image, if you see, only a trained and skilled pathologist can only infer. It's not so easy, understand? So but still we'll get a gist how what differences we have to look for normal and benign and for malignant slides, and that's what we will feed that thing in code in your Python code, and we'll tell the system to look for those patterns. Okay, so I guess I have covered most of this thing. What are histopathology images? Histo means tissue, pathology means study of disease. You can see those things, and they are the gold standard of cancer. Okay, what we do, whatever the part, okay, whatever the body part is suffering from, either, you know, it has either tumor or have a malignant cancer, the tissue part is cut off. What it does, your tissue part is cut off. Understood? And then they are prepared, they are fixed. Okay, one slide is taken, this is a glass slide like a finger length slide, and then with the help of a microtone device, they are sliced. So tissue is a 3D structure, your tissue is a 3D structure, and then we are slicing it into 2D structure, then we will be fixing it. Okay, then we'll be embedding it, we'll use some wax or something to embed to fix it here, and then we will use staining. So what we will use, so we will use eosin stain and hematoxylin stain. Understood? So these two stains. So eosin will color everything. Eosin will color everything pink, and hematoxylin will color everything as purple or blue. Okay, so whatever, so what things are being what are the parts that are being stained, we'll just look at it. You can go through these images and you will understand. Now first let me tell you why we chose breast and ovarian cancer. The first and the foremost reason is, like, most of the studies are done in breast cancer because it is the most commonly diagnosed cancer in women globally. Okay, and also it is widely studied, so their subtypes have been well defined, their signatures, as in their patterns. What are the patterns? So their patterns are used for also for cross cancer treatment decision, okay, but some patterns might be repeating, so they are used for other malignancy diagnosis also. Then complexity. Now, ovarian cancer, I have told you in the last class also, because it is very it is an inner organ. You cannot touch it, you cannot feel it so often. So it is detected very later in the stages. Okay, and that is why it is a quite contrast to breast cancer. But nonetheless, their subtypes have also been, histological, that is tissue subtypes, different types of tissue cancer like serous, mucinous, and endometrioid, clear cell. So these are different types of subtypes of cancer. They have also been distinguished. Previous studies have been done. Okay. Now, data availability is also easy for them. So we will look at them at 7th day. Those data set, I'll tell you later on. So what key takeaway you have to take from this class is, histopathological image is an image of a tissue which is stained. Got it? Everyone? Just react or just say something. Okay. So it is stained. I told you 2 dyes are used. Okay, so first is H, hematoxylin, and E, eosin. This will give purple to blue color. This will give pink color. Okay, and then we will look at those tissue slides under microscope, and we can see the tissue level architecture, like from far how it is looking. Okay, then we can zoom in that, and we can see the cellular architecture. So both things we can do. Basically what we are trying to say is different magnification. Okay, different magnification can be seen. So for AI term, what these images is our primary input data. Okay, for you to first understand them biologically is non-negotiable. Okay, that is why this class. Yes. So let us look at the second section of today's class, that is biological intuition. Yeah. So biological intuition. So reading a tissue slide, how to read that. Now you have got some tissue slide, how you will understand what's there in that. Only then we'll code for the AI to understand those things. So before you can train an AI model, you need to understand what AI is actually looking at. So first we will see what are the different hierarchy that we have already mentioned earlier. So hierarchy is different levels. Hmm? Now these are some levels. So the first and foremost level is the tissue architecture. So the overall organization, how the gland is looking, you just step back, take two step back and look at the painting from far how it is looking. Okay, no details now, just bigger structures like ducts are there or whether lobules are there or not. It will tell you, by looking at the bigger architecture, you can just say what tissue type is there or how those tissues are arranged in that organ. Then you have in biology, there's something known as stroma. Stroma is the nearby surrounding. If you see a gland over here, now if I want to show you stroma over here, yeah, you see. So here, this one is these are the glands. Okay, from here, these are the normal cells. So any secretion, it happens from these glands, and you can see these connecting tissues, these are known as stroma. So you should the computer vision, the AI must also look at the stroma, not only the gland pattern or the bigger tissue architecture. It must also have a look on stroma. Why, I'll tell you later. And then the after looking at stroma, you should also inspect individual units or cells. What are the shape of those cells, size, how they are arranged. Okay, and what are the diagnostic clues they are giving. Then deep, you dive much deeper. You look at the nuclei. So nuclei are the control center of the cell. So nuclear features also you should see. What are the nuclear features? I'll tell you. So it is one of the most important point to look after, for cancer grading, which stage the cancer has progressed into. Then you have got cytoplasm. Look at the cytoplasm also, right? Because there's something known as nucleus to cytoplasm ratio. All these we have to find. So all these things will be changed into numerical data. Understood? I'll tell you how. Normal versus tumor. So what are the changes? Now let us see. I'll not blabber, I just tell you. We'll just look at the screen and now we will read out, and then you will understand what will be the difference between a normal and the tumor cell, whether it is a benign one or whether it is a malignant one. Okay, so malignant is like last stage. Benign also, you can say benign is like maybe uncontrolled growth of cells, but benign are not always harmful. Understood? Benign sometimes can be harmless also, they can be slightly removed. Sometimes only these benign can go haywire and they can convert into malignant tissues. So now, normal or breast ovarian tissue, you will be looking at glands, ducts. They are very round, okay? They are regularly shaped and evenly spaced, nice arrangement. But if you look at any malignant tissue, the glands are irregular, mishappen. You cannot now you cannot recognize the shape of the gland, or maybe entirely absent, closed, not functioning. Cells are uniform in size and shape when it comes to normal tissue, but here every cell will be different from each other. And this is known as anisocytosis, when the size and shape they are different then nuclei are small and round. Now this is important. I told you there is nucleus to cytoplasm ratio. Okay, this ratio is smaller in normal cells. Please keep in mind, okay? This is an important point. So the nucleus to cytoplasm will be more and nucleus will be less. So the overall nucleus to cytoplasm ratio will be smaller. But in tumor or malignant tissue, you will have nuclei. They are enlarged. They are also irregular. Hyperchromatic. Hyper means more. Chrome means color. They'll be darkly stained because high DNA activity. Reason also you can see the DNA, more proteins and DNA are involved because it is rapidly dividing. Understood? The reason also you must know why they are hyperchromatic. They are all the machineries, all the energy, all the molecules are here because they want to divide so rapidly. That's why most things are stained and they are darkly stained. Now, stroma, the nearby surrounding connective tissue, they are organized and they're proportional to epithelium. Epithelium is kind of like, membranous cell. I'll not, this is something if you don't remember what is epithelium, you go to 9th standard science and you'll understand. Now, cell layer are one or more cell thick, no crowding, so they are finely arranged. If you see at the tissue, a slide, they are not too thick, one or two or maximum three layers of cell. The cells are not crowding. But here you see crowded cells will be there. Now you will here and there, I told you why you have to look for stroma, the nearby area, because here and there you will have mitotic figures. Mitotic, that means cell division, because they are caught dividing rapidly. Again, the same point. Stroma will be dense. Now, this is known as desmoplasia, where the stroma is very dense. Again, why? Because all the biomolecules are here because they want to help cancer cell to divide more. Okay, and it is a response to cancer. So here you have your immune cells, and fast dividing machineries. All those things will be clumped at stroma. So stroma will be dense. Cells invade the surrounding tissue. I told you it won't only affect itself, but it will also damage the nearby tissue. So it won't respect boundaries, and only later on it will leave its own place and spread throughout the body because there is no respect for boundaries. So, very fast, I'm telling you, there is a topic in CSIR net. Actually, why I'm quoting it, because you have to go through this once. Even if you're not giving the exam, please go through the syllabus once. It will be very helpful to you. What happens, suppose a cell is growing and dividing and it is coming in contact with the next cell, it will come to know now it is the time for me to stop growing. But cancer cell will not respect that boundary. It will increase in size, it will invade other places, it will destroy the nearby cells also. So you got it? Till now, what I have taught you, did you get it?
Yes, ma'am.
Okay. Do you want me to repeat anything? Anyone else? Till now? So, as a biologist, if we are entering into computational biology, please don't leave your biology behind. we have got one data set. So there we have various magnification levels. Okay. So in break his data set, it is a kind of data set where you have 40x magnification, then you have got 100x magnification, 200x magnification, and 400x magnification. And what is magnification? So why anybody has ever handled a microscope? Anyone? Normal school level or college level, have you ever handled one?
Yes, ma'am.
May I know the
Yes,
name?
ma'am.
Harsha? Yeah.
Yes, ma'am.
Kiran? Very good. So what
Yes.
you do? Yeah. What you do first, whenever you set a slide in the microscope, first, in normal compound microscope, you have 5x or usually you have 10x, first objective lens. First, you in a lower magnification, you set to get a field of view. After the field of view is set, then you slowly increase the magnification to look into the details. Yes? Am I right?
Yes, ma'am.
So these different magnifications, it will give you different information or different data. So what you see at the lowest level magnification, you will just see the overall architecture of the tissue. You can see the glands, you can see the ducts, the overall, what shape they are forming. The stroma is visible. These lightly colored neighboring connective tissue, the stroma, that is visible. Okay, but the cellular detail is not visible over here. Then you slightly increase the magnification. At medium magnification level, you start to observe the individual cells, okay, and maybe cytoplasm, and roughly you can see the dark portion as the nuclei. And at a very higher magnification, you can further see the details, like patterns, how chromatins are. And you see here the purple patches, like mitotic cells also is visible. Okay, all those things you can see, right? So as we increase the magnification, we can see more details. I have a question for you. Can you tell me, for computer vision, for CNN classification, is higher magnification the better or not? Or is lower magnification the better or not? Any guess, wild guess?
Higher magnification is better.
Hariam was telling higher magnification is better. Anyone else?
Maybe lower.
Afrin is telling lower. Fine. Can I tell you what did we find when we were working on this project? Yeah, no, yes, no, no. Okay, fine. No problems. So,
No.
what did we find is in break his, I'll break it to you now. So, it is like 100x, 200x, and 400x. The most poor performance given by the model was 400x and 40x. And the most optimal magnification for break his was 100x. Okay, what happens, the reason what we found through literature search or through our own intuition was like, so the deeper we go into the cellular structure, so it only gets one detail. What kind of detail? Like, what is the pattern of nucleus and what is the shape of the cell. And when we go to the very low resolution, we can only see the overall duct size. But if we are into somewhere in the middle of the magnification, like 100 to 200x was better performing. Why? Because we got not only the overall pattern of the gland or the tissue, but yeah, to some extent, the cellular pattern was also visible. Got it? Make sense?
Yes, ma'am.
Repeat again.
Yeah. So it makes
Not
sense, right?
so.
It is common sense, right? So we can see both at the mid magnification level. Yes, even I was thinking the higher the magnification, the better the result. Even I was thinking like that, but I badly failed at 400x. When, and obviously there were other reasons also. Initially I learned from my mistakes, so I'll tell you the other mistakes later on. But yeah, magnification also plays a very important role. So now whenever, if there is when you become a computational biologist later on working with images, tell your labs or your doctors to give multiple magnifications so that you can feed those magnification and check whether which model is being trained well in which level of magnification. It will come somewhere in middle, not lowest and not the highest also. So moving on, benign and malignant. So the critical decision, why? Because this only our CNN model has to make. Now, what it is telling, not every abnormality is a cancer. No. Okay. So it is a learnable, visually learnable distinction that you have to learn it. Just you see as abnormality. See, why I'm telling you why all abnormality is not a cancer, because you can have human errors also. You would have taken completely normal tissue, you could have smeared it, and you could have disrupted the morphology. Understood? So not every abnormality is a cancer. For benign for benign tumors, what you will look cells, it will be more inclined towards the architecture of normal tissue. But in malignant, it will be more atypia, like they are abnormal and not of same types. Then benign also, they will stay within their boundaries. They will, then why they are benign if they are resembling so much like normal tissue? The only reason is it is lying inside the nucleus, the biochemical processes and mutation. Okay. So biochemical processes and mutations are taking place right now inside them. It might be hazardous in future, or it may not be also. So that's why the morphology is mostly like normal tissue till now. It might be slow growth, or after a point of time it can stop growing also. But yeah, once it has turned to malignant, it is rapid and uncontrolled growth. Now, benign will be kind of a local mass. It won't spread to distant site, but malignant will spread. Okay, it will reach your blood and lymph, your liquid tissues. Okay, and then it will spread to your body, and this process is known as metastasize or metastasis. So metastasis is a noun and metastasize is a verb. Okay, some examples are given. Fibroadenoma is a benign lump in the breast which can be removed and you're good to go. But if you have a ductal carcinoma, so carcinoma, you see, it is cancer, malignant. Okay. So with the help of, if you learn and give time, there'll be a point of time whether by looking at the name you can try to understand whether it is a benign or a malignant tissue. Now, suppose there is a trained pathologist. What cues it must look for? First, it will look for pleomorphism. Okay. So morph means how does it look, how the looks are. So the size and the shape of the cells and nucleus will not be normal. Okay. They might be 5 times larger in size than the other cells around them, from the benign or the normal cells. Then there'll be architectural disruption, like there will be glands will not be visible because everything has been disrupted, right? Solid sheets of cells, why? Because they are growing one on the top of other uncontrollably. So solid sheets of cells, they will pile up at one place. So these all are the signal for malignancies. Okay, so but if you go for the normal architecture, they have a predictable pattern. They will have a round gland and all those things which I have already discussed earlier. Then if you look at the cellular density, the tumor regions are often hypercellular. More cells are present in tumor region when you compare to normal region. Okay, so AI model will look at these things. Then you have also got mitotic. Okay, I'm sorry, I just forgot. Just give me a second. I had forgotten to send a class link to other people. Okay. Okay, fine. So these three things I have already told you. Now, this also I told you. I'll tell you later on why to look for mitosis, mitotic figures. Now, mitosis is cell division. Okay, mitosis is cell division. Let me check whether he has joined or not. Can you see my screen now?
Yes, ma'am.
Hariharan, have you joined?
Yes, ma'am.
Sorry, Hariharan, I forgot to send. I sent it to everyone, but I forgot to send it to you. You can just go through the recordings later on. Okay, we'll move further. So, what actually, just to give you a gist, I told you something about how magnification plays an important role. Okay, Hariharan? I told about
Okay, ma'am. I'll
magnification.
refer to the video.
Sorry.
Okay, ma'am. I will refer to the recording and
Yeah.
send that
Okay.
again.
So I'm
Mm.
just revising that I've talked about magnification, and now we are going to look at these what a pathologist must look, okay, for cancerous pattern. So it is pleomorphism, that is irregular shape. Architecture is totally disrupted. Then near the tumor area, the cell will be more dense because you have more cells nearby. And then we are here in mitotic figures because since it is a fast actively dividing area, so cells are caught dividing over here, and high mitotic count you will see. High proliferation means it is division. Okay, and then you can say that the more number of mitotic figures you see, it is more aggressive tumor. So you can also help grading of the cancer, at which stage it has gone. Okay, now nucleus to cytoplasmic ratio also I have discussed. So n, nucleus is small in normal tissue and cytoplasm is larger area, and in cancerous tissue this nucleus to cytoplasm ratio increases. Okay, fine. So these things you must know. So moving on further, so visual anchors, I mean, what visual cues you have to look for? What are those things? So, what is the shape? How they are spaced? And what is the size? Whether they are irregular or not. Okay. Then you have to see the nucleus size. Are the nuclear size same or varied? Okay. And how they are stained. Okay, whether organization is normal or they are haywire here and there, chaotic, whether they are dense or whether they are sparse, and whether they are confined to a local space. So when they are confined, they will respect the boundaries. Okay, so they will respect the boundaries, and when they are invasive, they are metastasizing. Okay, so these are the things you have to understand. Fine, these are the things you have to look for, whether they are regular or irregular, whether they are uniform or they are varied cells, whether they are organized or whether they are chaotic organization, whether they are loosely packed or sparse or whether they are dense, whether they are confined at one place or whether they are invasive. Okay, all these things, these are the patterns you have to look for in a histopathological image slide. Now, let us come to the AI part. You have seen the histopathological biological part. Let us see the AI part. So, what AI does is that AI model actually, what do they process? They will process the pixels. From pixels, they will look at certain patterns and they will try to generate some and try to learn from what are the features. I'll tell you what are these later on. And when they have learned from all these things layer by layer, neural networks, I told you, neural networks, they work in layers. So layer by layer, one thing at a time, they will learn. Okay, so at that time, when they have learned everything layer by layer, then they will go for predictions. Understood? Then, so an AI model does not see nucleus. As a human, we see nuclei. They are round, whether the glands are present or not. Okay, it will only look for numbers. So they would not look at raw images. We have to process them. Okay, we have to process into different data type. Can you keep this thing in mind, everyone? I want a yes from everyone, please.
Yes,
Data
ma'am.
type, a
Yes,
different
ma'am.
data type, not image. We will give the image, you will not convert it. By coding, the model will itself convert them. Okay, so don't worry, you don't have to. When coding, you will understand that, oh, okay, it's not that tough also. Everything the code is going to do for you, the packages will go, pre-built packages is going to do for you. I'm just saying this happens. Raw images will be converted into different data types, and these data types are known as tensors. Have you heard about these things? Tensor?
Yes, ma'am.
Yes,
Yes,
ma'am.
I will show it to you. What are tensor? First, let us finish with this chunk of information. Now, each pixel, so each dot is just a value between 0 and 255. And these are intensity of colors. Intensity of color. Okay. I'll just stop share and I will show you. Wait. Now you will understand. Can you see an MS Paint window over here?
Yes, ma'am.
I'm going
Yes,
here
ma'am.
and I'm mixing my color. Can you see here RGB? Red, green, blue. Okay. So, and you see there are 3 fields over here. So, and here the value of the color will be converted into numbers. Can you see here? This one? Let me annotate. Yes. This area is the numerical value for the color. Okay. In MS Paint, everybody you would have drawn any image in MS Paint, but you would have never thought about it like this. So any color is divided into 3 channels: red channel, green channel, blue channel. So red value will be from 0 to 255. Green value will be ranging from 0 to 255. And blue value will be ranging from 0 to 255. And by the combination of all, we have got nearly 256, near to that 256 million different combinations of colors we can generate, with this. Understood? I can also show you now. Suppose if I
Yes, ma'am.
type 255, you see and the green is 0, blue is 0, I'm getting a pure red. Can you see over here? And if I cancel this red, and if I write 255, you can do it by yourself also in your system. And 255 here, this is pure blue. What if can you tell me what color I'll get if I mix all the colors? Now I'm mixing red and blue, I'm getting pink or
White.
so what color will it get if I mix everything?
I guess white, ma'am.
White color. You don't guess, you get white color. Everything is reflected back, so it's white, right? Color mixing by addition. This is known as color mixing by addition. But if you try to replicate this in paint, if you mix RGB in painting, you will get black color, not white color. So that's color mixing by subtraction in paint. And when it comes to lights, it is color mixing by addition. Okay, I'll stop share and I'll share my website page again. And you see, I hope you got a little bit of understanding what are those 3 channels that we'll be dealing with later on. And each pixel will be having 3 channels: red value, blue value, and green value. Understood? Now, each pixel is just a value between 0 to 255, and it will represent the intensity of the color. A pixel is nothing but, single pixel, it will not have any meaning. But a cluster, many pixels together, when they are going to form a shape, that is a pattern, and that pattern will have some biology. If it is how it is biology? If that pattern is circular, it is a normal gland, it is a normal tissue. If it is very irregular like this, it might be tumor, or it might be malignant or benign. Understand? Usually benign will be roughly round only, but if it is very disorganized, it will be malignant. Yeah. So by studying the group of pixels, suppose if those pixels are having high intensity of blue and purple stain. So blue and purple, either B value will be more, and if it is purple, it will be red plus blue value if it is more. That means it will understand here more of the hematoxylin stain has been absorbed. And that channel might be, those structures which have the tendency to absorb more stains. So nucleus, usually they tend to absorb more stains because they have negative charged molecules. Please note it down, this is also very important. So hematoxylin will stain mostly negatively charged molecules. So DNA, RNA, and histone proteins are also, no, not the histone proteins. So DNA, RNA are the negatively charged. There are some other negatively charged proteins also that they are negatively charged. So it will bind, those stains will tend to bind. That's why nucleus appears purple or blue. Okay. So the model will learn with the help of pixels. It will learn this association. It won't learn the biology. It will learn only pixels. Then if we zoom out at a particular region, spatial repetition, spatial, yeah, it is coming from the word space. Okay, in a particular space, in a particular region, how these nucleus are clustering together. Okay, if more nucleus, that means more cell. If more cell, it means the area is more dense, and we have learned that it is a malignant region. Okay. So the model will learn spatial statistics also. So we are zooming on from pixel to local pattern to regional pattern. Okay, we are zooming out. And then finally it will look at the complete global space. Now, if it has seen high cellularity, that means if it has seen dense area, and if it has seen that the architecture is disrupt, the glands are not found correctly, okay, the nucleus is irregular, then it will predict high probability, okay, high probability that it is tumor. In that also, if it is more inclined towards normal, then it can be benign, or it can be malignant. Okay. So layer by layer, the convolutional neural network will try to combine the features, and then they will try to learn. So moving on further. So this is just an illustration, you see. So pixel, here there are some light area, light pixels, dark pixels. Here you can see more dark pixels. So in first layer, it will try to learn the edge. Then it will learn the overall shape. Then it will see the regional patterns. Then the overall structure. After that, in the last layer, it will try to assign what class, what different type of tumor it is. Okay. Also, not only, like I told you, it is not binary classification, not only normal versus tumor, but if it is tumor, what kind of tumor it is. Then it will generate a feature map, which we will see in the next class, and then it will assign probability. So every classes will be having a probability, but it will choose only those class which has the maximum probability, but we will learn this in the next class in detail. Okay. Now, so why it is this hard? This is not a simple problem. It is hard for biologists also, for computational biologists also. Why? First of all, in different labs, even if they might be using different chemicals from different batches, but suppose even if you're using same chemicals, every time you might stain the slide differently. The intensity of the same tissue might not be same. The same tissue type can appear purple dominant in one hospital and pink dominant in other hospital. Understood? It is very simple language. Understand? Did you get it? Because it is
Yes,
human
Yes,
error.
ma'am.
This is how humans are handling things, right? Now, a single intensity value change of 1 to 2, from 0 to 255, even a change of 1 from 0 to 5, five-fold change also, it can bring difference in the computational results. Now, this is a batch effect. When human error comes, no, it is a batch effect. Batch effect. It is known as batch effect. Very important. So you will encounter batch effect in every bioinformatics work, not only in this computer vision problem, but everywhere. So it is a major challenge for AI generalization. Then you also have got biological variability, because cancer itself, if it is from the same lump, it is not uniform. Two slides from the same tumor can look dramatically different. Within the tumor, intra-tumor heterogeneity also is there. Hetero means different types. They are not uniform. Tumor within the same lump are different. That is why heterogeneous. And that is why the classification is difficult. You cannot expect 98, but you can get many papers out there. They're saying 97% accuracy. I don't understand how. Okay. So we got 75% accuracy and we were happy with it, but not as happy. So we are proceeding with our project. But still, it is a very good percentage for now, 75%. Because, see, we are dealing with heterogeneous cancer. Now, also scale dependency, we have different magnification level also. Okay. And then class imbalance. So this is something challenge, like for each and every subtype, you don't have equal number of images. Some classes, like in his data set, breast cancer data set, there are 8 subtypes. Okay, 8 subtypes you have. In one, you have got 5,000 images. In another, you have got only 135 images. So don't you think that the subtype which is having 5,000 images will be dominating the prediction mathematically? Because the number of the images are more. Do you understand? You're feeding more images, more numbers. So what they will understand, that you will understand this in when I discuss the data set. But for now, you just remember that more, if class is imbalanced, what AI will understand is, okay, its prediction will lean more towards that class. There are 100 images there, and there are 2 images here. It won't learn from the class where it has 2 images. That subtype, it won't try to learn from them. Class imbalance, if it is there, the more population of images will be dominating the AI model. Is it okay? Now, do you understand a slight idea? Have you got it?
Yes, ma'am.
yes, ma'am.
Yeah, so that is also a problem. So here the computational checkpoint. So when you look at the slides, I'm giving you a homework now. We'll discuss it later on. So when you look at the slide, what type of numerical properties can you get from CNN? So if suppose there is a dark nuclei, what numbers you will get? From 0 to 255 pixel intensity, from here one number you will get. Yes or no? You got one number? Yes? See, the
Yes,
nucleus
ma'am.
is darker than the surrounding. So the pixel intensity that I told you, know, that 0 to 255 value will be more or not?
Yes, ma'am. It will
Yeah,
be
so
more.
we have extracted one number so that the AI will understand. Okay. And if there are crowded cells, obviously this is like crowded cell means malignant cells. So high local pixel, there will be more number of pixels. Yes or no? And
Yes, ma'am.
background will be less. The background pixels will be less because more cells, more cellular pixels, less background pixels. Again, so pixel ratio, we are getting this in the ratio. Again, we have got another number. So with these number, our CNN model will learn. Now suppose irregular shapes, how the numbers are coming out from shapes? It may assess the perimeter to area ratio. Now did you get? Numbers will come from these things. Yes? And if something you understood, you can handcraft features from your side also. They will try to remove this CNN features, no? It will try to learn by itself. CNN, this AI model itself will try to learn features. You don't have to work. You just have to pre-process the data and give it to the model. That's it. So 3-level scan protocol. What is it? We'll just go fast now. So architectural scan, step by step, you have to see first the whole tissue. Then move closer, look at the cells, cellular level cell population. Okay, and then zoom inside the cell and see whether inside how the chromatin is stained, how they are dividing. So these are the 3 levels. Okay, you have got the link and you can go through this page again. Now, once you have seen a tissue, these are the things you have to check for. What is the density? Whether it is regular or not, whether the cells are uniformly arranged or not, whether it is respecting the boundaries or not. So all these things, repetition, necrosis. What do you mean by necrosis? It will kill, cells will die, it will kill the healthy cells. Healthy cells are killed. Necrosis, death of cells, uncontrolled death of cells. Understood? And at that place, you will not get a visible nuclei. Only cancerous nuclei will be there. Other normal nuclei will not be present. Fast dividing, so you should go for mitosis also. Overall impression, what you are inferring, that also you have to understand. Okay. So what are the common beginner's mistake? Don't treat it like any other image. Why you should not treat it like any other image? Because you see, it's like 100,000 or 1 lakh by 1 lakh, 100,000 by 100,000 pixels. So there are so many domain shift, colors, spatial context, many patterns. Never ignore the biology. That's why I'm telling you this foundational class is very important. Never straight, don't deep dive into deep learning. Never. First, understand the basics. Only then you will, otherwise, you will never be able to debug what is the problem. Label, this is very important. When we will, at day 7, I'll tell you when we will get to the overview of the data set, ignoring every folder, every patient name, every type of cancer subtype is labeled. Names are given to each folder and images. And this is playing the most, please note down, most important role. Names are there, only then it will classify. This is a classification task. Only with the help of names it will classify. Okay. All these things are present in our data set. They never forget about the magnification also. So this also I have discussed already. So why this matters for CNN? This is redundant. You can go through it. So what is the big idea? So the big idea is computational pathology researchers are not the best coders. Not you, neither I am the best coder. Okay, but we are researchers. We can move fluidly between biological meaning and computational representation. Okay, now you understood? This will give you some confidence, right? Right?
Yes.
This is some assignment. Try to do it. Okay, you have Monday to Friday. You can go through this. So this is an observation table. I will be sending via mail some images, and just look at two images from each subtype and try to fill it, whether you can understand which image is showing you normal or malignant. I'll just communicate with you later on within the upcoming week, and you please try to do this assignment. Okay, if you don't understand, you can contact me also. So this is the outcome summary. You can go through that also, and tomorrow we'll be discussing the convolutional neural network basics. Okay, so was the class helpful?
Yes, ma'am.
Yes,
Yes,
ma'am.
ma'am.
Did you
Yes, ma'am.
understand?
Yes, ma'am.
Okay. So it's okay.